Good afternoon, and welcome to the UCSF Academic Senate's 63rd Annual Faculty Research Lectureship in Basic Science. It's my honor to introduce the Academic Senate Faculty Research Lecture. My name is Margarita Lightfoot. I'm a professor of medicine in UCSF School of Medicine. I'm the chief of the Division of Prevention Science, and I direct the Center for AIDS Prevention Studies and the UCSF Prevention Research Center. And I also chair the Committee on Research. This award recognizes exceptional career achievement in clinical science research. The Senate has two other faculty research lectures, one in clinical science and one in translational science in the spring. Nominations are made by UCSF faculty who consider scientific research contributions of their colleagues and submit nominations for this prestigious award to the Academic Senate Committee on Research. Every year, the Committee on Research selects a recipient for this award. I'd like to thank the committee for their hard work. Today, we are proud to honor our colleague, Kevin Shannon. Dr. Shannon is a professor in the Department of Pediatrics. Professor Ophir Klein will introduce Dr. Shannon. So I'm pleased to turn this over to Professor Klein.
Thank you, Dr. Lightfoot. So on behalf of my co-nominators, Drs. Ben Braun, Mignon Lo, and Jeff Feynman, I'm delighted to introduce Dr. Kevin Shannon, who is the Roma and Marvin Auerbach Distinguished Professor in Molecular Oncology, as the recipient of the 63rd Annual Faculty Research Lectureship in Basic Science. Kevin is a pediatrician who has made seminal contributions to our understanding of cancer and developmental disorders through his research on aberrant RAS signaling. He has defined causative mutations, elegantly characterized their biochemical and functional properties, and engineered innovative and accurate in vivo models that have provided novel insights into mechanisms of response and resistance to anti-cancer drugs. I'd like to tell you a little bit about Dr. Shannon's amazing career. Early on, Kevin was a key member of the research team that pioneered the use of erythropoietin in premature neonates to reduce the need for blood transfusion. And in the mid-80s, Kevin and his colleagues noticed a profound immunodeficiency emerging in previously healthy neonates and suggested it was likely associated with exposure to blood transfusions. A manuscript describing this observation was initially rejected for being implausible. Before long, the development of AIDS in hemophiliacs clearly established AIDS as a bloodborne disease. However, this might have been known sooner had Dr. Shannon's initial manuscript been accepted. After performing his fellowship in pediatric hematology oncology at UCSF, Kevin worked with YW Kahn while on active duty service in the US Navy. As a fellow, Kevin became interested in the role of monosomy 7 in childhood myeloid malignancies, such as acute myeloid leukemia and myelodysplastic syndrome. He identified several families in which multiple siblings developed AML with monosomy 7 and banked samples from them. He helped to establish how monosomy 7 carries a poor prognosis in AML and to narrow down the relevant region on chromosome 7. More recently, as he'll tell us today, the use of these valuable primary specimens allowed him to solve a 30-year-old mystery about the origin of these cancers that are of major significance to pediatric oncology. Later, in a seminal paper, Kevin performed genetic studies in families with neurofibromatosis type 1, one of the most common autosomal dominant genetic disorders in children. He demonstrated that tumors arising in NF1 patients undergo somatic loss of heterozygosity, suggesting that this condition followed the tumor suppressor model analogous to familial retinoblastoma. Kevin is perhaps best known for his pioneering work defining juvenile myelomonocytic leukemia, or JMML, as fundamentally a disease of hyperactive RAS signaling. And he went on to generate and characterize the first animal models of this aggressive childhood cancer, which he deployed to perform rigorous preclinical trials of targeted inhibitors. These bench-to-bedside discoveries are being translated into clinical practice through a novel multi-institutional, pathway-focused National Cancer Institute-funded SPORE program. Kevin also discovered germline KRAS mutations as an unexpected cause of Noonan syndrome, and he functionally characterized these novel alleles. Most recently, his lab developed strategies for testing responses to anti-cancer drugs in vivo and characterizing mechanisms of resistance. His seminal studies of inherited pediatric cancer syndromes and developmental disorders, his mechanistic contributions elucidating how aberrant RAS signaling causes human disease, and his efforts to test new therapies make Dr. Shannon a truly exemplary physician scientist. In addition to his outstanding research program, Kevin has had an important administrative career in the service of science and medicine. Over the course of his time at UCSF, he has held numerous positions in which he has made extraordinary contributions. Kevin served 2 tours of duty as Pediatrics Department Interim Chair, and he also has served for many years as Vice Chair of our Department of Pediatrics, as well as Director of the UCSF Medical Scientist Training Program. He has held key national leadership and advisory positions, including service on the NCI Board of Scientific Advisors, and has chaired the external advisory boards for leading programs such as the Research Institute of the Hospital for Sick Children in Toronto and the Memorial Sloan Kettering Center Human Oncology Pathogenesis Program. And he is a member of several other boards, including at the University of Pennsylvania, Abramson Cancer Center, and the UC Irvine Cancer Center. Throughout his career, numerous honors and prestigious awards have recognized both Kevin's science as well as his leadership. A sampling of these includes the Samuel Rosenthal Prize for Excellence in Academic Pediatrics in 2004, an American Cancer Society Research Professorship in 2010, and election to the American Association of Physicians in 2010. More recently, in 2019, Kevin was awarded the prestigious Society of Memorial Sloan Kettering Prize, given annually to an individual who has made significant or groundbreaking contributions to the field of pediatric oncology. Kevin has taken an active role in mentoring numerous individuals who study areas outside of his primary interests as well. He has influenced the lives and careers of numerous medical students, residents, fellows, junior and senior faculty, and colleagues in the United States as well as abroad. I want to close with words about Kevin's mentoring from the letters that my colleagues wrote. Ben Braun wrote, "Kevin is known widely not only for his expertise, but also for his genuine commitment to partnership. Time and again, scientists who made initial observations have turned to Dr. Shannon for advice and collaboration in order to study the larger impacts of their findings." Jeff Feynman wrote in his letter, "Throughout my, as well as many of my colleagues' careers as physician scientists in pediatrics, Kevin has served as a selfless mentor and advisor. As interim chair of the Department of Pediatrics and director of the Medical Scientist Training Program, Kevin's devotion and commitment to mentoring trainees and junior faculty has been truly impressive. Simply stated, Dr. Shannon is one of the few remaining quadruple threats in medicine. He is a superb clinician, teacher, scientist, and administrator." Mignon Lowe added, "Kevin Shannon is the ideal physician scientist. He is thoughtful and inquisitive, curious about topics beyond his laboratory focus, always asking pertinent questions in a straightforward and intellectually rigorous manner. There is also a seemingly paradoxical quality about Kevin. Most who know him appreciate that he certainly has some very strong opinions and is not shy about sharing them with you, whether you like it or not. But at the same time, he is one of the most understanding and supportive colleagues I have ever had the pleasure of working with." On that note, please join me in congratulating Kevin on his extraordinary accomplishments. Kevin, we're all looking forward to hearing your seminar today.
So thank you, Ophir, for those really overly kind words and also for giving a great introduction to what I'm going to be talking about today. It's a really distinct honor to receive this award from my peers, and I'm deeply grateful to the Academic Senate for giving me this opportunity. So I wanted to acknowledge a couple of people from the start and in a way dedicate my remarks to them. The first is Zena Werb, one of the really wonderful faculty members at UCSF, a terrific scientist, great mentor, and an all-around mensch, wonderful supporter who we lost tragically this year and unexpectedly. And Zena was the 44th faculty research lecturer back in 2000. And then my own mentor, who you mentioned, Y.W. Kahn, who was the 27th faculty research lecturer back in 1983, a pioneer in the genetics of hemoglobinopathies, sickle cell disease, prenatal diagnosis. He essentially invented molecular prenatal diagnosis and was the most generous mentor anybody could have had during my fellowship training. I'd also like to start by giving special thanks to three colleagues who have advanced my careers in so many ways and have worked on so many different projects that it's impossible for me to even think about giving a lecture like this without their friendship, support, and collaboration. The first is Michelle Lebeau, who I met in the late 1980s when I at a children's cancer group meeting, and Michelle is the world's foremost expert in secondary leukemia and in chromosome 5 and 7 deletions in leukemia, and encouraged me to continue to work in that area, collaborated with me, and with her colleague Janet Rowley, invited me to join a program project grant at the University of Chicago that I was in for 15 years that allowed us to continue this work. And I've known Michelle now for well over 2 decades into 3, and she's the kindest and most generous scientist I think anybody could wish to have. Second is Tyra Jacks, who is a graduate of went to graduate school at UCSF here, where he worked in Harold Varmus's lab. Tyler is currently the director of the Cancer Center at MIT, a National Academy member, as many of you know, and a pioneer in developing genetically engineered mouse models of cancer. And Tyler's major interest is in the area of epithelial malignancies. And so as he developed these pioneering models of RAS cancer, he generously shared them with us and with my laboratory to characterize them in the hematopoietic compartment. And much of what we've done over the last 2.5 decades has been attributable to the wonderful friendship and support of Tyler. He's family to me, as are Michelle. And the third person I'm going to mention here, Gideon Bolag, who I met when he was a postdoc in Frank McCormick's lab. And Gideon is an expert on RAS GTPases and biochemistry. And the focus of my lab has always been to try to link biochemistry with the ability to do biochemistry in primary cells, which is sort of doable in the hematopoietic compartment, but not easily doable, and would have been impossible without Gideon's advice and collaboration. And so you'll hear a good deal about all of their work. For the younger people in the audience, I would just mention that I looked back at my CV yesterday, and I've published over 10 research papers with Michelle, Tyler, and Gideon over the years, including some of the most important work from the lab, and that work has spanned over 2 decades. And so one of the wonderful things about science is when you meet kindred spirits who are generous and talented scientists, they can advance your career, and you can hopefully make a small contribution to theirs. So what I've decided to focus my talk on today is to talk a little bit about different paradigms of how we do discovery in an academic medical center like UCSF. And this is, of course, an oversimplification, but I'm going to just kind of go with it and ask you to go with me. So if we think about the basic science paradigm, we think about well-trained and talented creative individuals who come up with a novel question and do an experiment, and then make a discovery and then pursue the next question. And this obviously often involves a great deal of innovation in terms of inventing novel technologies to address these questions and staying on task and on a question and focusing deeper and deeper, peeling the skins off of the onion, whether that problem is something like the unfolded protein response or how organisms solve the end replication problem at telomeres. The physician scientist, what I'm calling the dilemma, is somewhat different. When I refer to physician scientists here, I'm talking broadly about physicians like myself who don't have PhD degrees, and also MD PhD degrees, MD PhD colleagues who engage in patient care. And here I think a lot of the research is informed by clinical observation. And in many ways, what our job is to try to understand what our patients are trying to teach us. And it's that clinical observation that that brings us to do an experiment in the laboratory. And if we're fortunate, we'll make a discovery. But I think at that point, we often are confronted by three different sort of challenges. The first is there a next lab question, and can it be addressed given existing technologies? And in my own career, I've run into at least one clear example where we made a discovery and just couldn't move it further just because the technology wasn't ready. And I'll talk about that in the next section. The second is these patients have informed us about their disease and given us a great gift. Don't we owe it to them to now try to take that discovery and bring it into the clinic? And then finally, what about something different? After we've looked at one disease and made a contribution, some new knowledge, can we then move on to study another disease? And I think this part of it is often sort of informed by the fact that we're constantly seeing patients in clinic, trying to connect the dots, and in some ways perhaps wavering from the original path to wander off into doing something different. And I'll talk some about this kind of a pathway and how it may inform what's happened to me for 3 decades or so. So just to tell you about myself, I was an English and biology major at Williams College, and then went to medical school at Cornell, where I was very interested in patient care. I matched in residency at UT Southwestern in Dallas, and was a general pediatrician at the Naval Hospital in Oakland until 1985. From 85 to 88, I was a fellow in hematology oncology here with YW. Then went back and finished my naval service and joined the UCSF faculty in 1992. This is my medical school yearbook picture, which my mother never liked very much. Shows me being a little bit scruffy, and also in the scruffy subway stations of New York City back in the late 1970s and early 80s. And this is what I looked like back a few years later, about during that general pediatrician time back in the Navy hospital. As Ophir mentioned in his very kind introduction, during my residency and time as a general pediatrician in the Navy, like many physicians who wind up doing research as part of their careers, it initially was founded by case reports. I then did a small lab project where we were trying to use 3 new antibodies called OKT3, OKT4, and OKT8 to the markers that are now called CD4, CD8, and CD3 in T cells. And I was interested in children with low platelet counts or thrombocytopenia, and stumbled into one of the initial cases of pediatric AIDS through that work, and was inspired to do this by George Buchanan, a wonderful clinical investigator at UT Southwestern, and later by Art Allman here at UCSF. And during my residency, after my residency, when I came out to the Bay Area, I was fortunate to be assigned to the Naval Hospital in Oakland and to the branch clinic in Treasure Island, where I met Harold Koenig, who was a hematologist in the Navy and also the chairman of our department. And he had learned from Connie Eves some assays in cloning and growing myeloid and erythroid progenitor colonies to study hematopoiesis and methylcellulose. And he taught me those assays, and I applied them, as Ophir said, to the anemia prematurity. And then that resulted in recombinant erythropoietin trials that I was fortunate to participate in with Bill Menser from our hematology division, and from Rod Phibbs from neonatology. And just parenthetically, this problem of anemia prematurity also inspired another pediatrician named Greg Semenza at Johns Hopkins to study why it was that preterm babies didn't make erythropoietin. And as many of you know, Greg shared the Nobel Prize last year for discovering HIF. He also graduated from Sleepy Hollow High School in Tarrytown, New York, 1 year before one of my brothers. So what I primarily focused my career on have been these myeloid malignancies, and they come in 3 flavors for those of you who are not hematologists. There are a group called myeloproliferative neoplasms, where there is an increase in cell number, but differentiation is fairly normal. And these disorders are mostly characterized by chronic myeloid leukemia, the disease caused by BCR-ABL. A second group is a more mysterious group called the Myelodysplastic Syndromes. And in this case, there's a decrease in the number of cells and also very abnormal differentiation and in the bone marrow. And finally, there's acute myeloid leukemia, in which there's both an increase in overproliferation of cells and also aberrant differentiation. And both myeloproliferative neoplasms and MDS will progress to AML as they acquire additional mutations. So as Ophir mentioned, the first problem that I studied in the lab with YW was this problem of monosomy 7 and deletion 7q. And these are very common genetic alterations in myelodysplastic syndrome and other myeloid malignancies. And they tend to be associated with adverse outcome in both pediatric and adult patients. There are some inherited predispositions that predispose to this. Aplastic anemia does, and prior exposure to genotoxins are known risk factors for developing MDS with monosomy 7 and a deletion 7q. And I got interested in this problem, again, typical of a physician scientist, by caring for a 7-month-old boy with MDS and monosomy 7 during my fellowship. And I remember telling his parents that after we did his bone marrow that we didn't know what he had, but he didn't have leukemia, and then having to come back a couple of days later after the cytogenetics returned to unfortunately have to tell him he had something much worse, which was this monosomy 7 syndrome. And some rare cases of MDS or AML with monosomy 7 had been reported in normal siblings at about that time. And it was YW who said, you know, you really, if you're going to try to do molecular biology and study inherited cancer predispositions, you need families. And I was able to pass his test by identifying 3 families to begin to study. And this is a paper from a very long time ago. I believe it's 1989. And we used the few restriction fragment length polymorphisms that existed at that time. And you see that these families are pretty characteristic. Both parents are phenotypically and hematologically normal. 2 full siblings, and they can be of either sex or they can be of the same sex, that develop myelodysplastic syndrome or AML with monosomy 7 generally during the first decade of life. And as we study these families, we propose a simple hypothesis that there was a mutation somewhere on chromosome 7, likely on the long arm, because that's the arm that's deleted in some myeloid cancers, and that children would retain the same parental chromosomal homolog. In other words, the one that harbored the abnormal mutation. And then the second hit, if you were, was then the loss of the normal chromosome 7 homolog. And we immediately discovered that this was incorrect. And I just to box this symbol family 3 out, using the 5 or 6 markers that were available on for chromosome 7 Q22 at that time. And we were fortunate there were this many, and it was because the cystic fibrosis gene is roughly in the same neighborhood. What you see is that these 2 sibs, who are both affected, have now a single chromosome 7 homolog, so only one of the 2 polymorphic markers. And you can see that they're different throughout this entire region for these 2 sibs. So again, did not prove our hypothesis. And we then became interested in more broadly in these chromosome 7q deletions and reasoned that they might be functioning by haploinsufficiency. And this has turned out to be true. And Michelle Lebeau had described this commonly deleted interval, and in subsequent years, her lab and others described other commonly deleted intervals within this chromosome 7q22 region and different candidate genes. And so to sort of tell you what happened between 1992 and 2016, the cytogenetic and FISH analysis of myeloid malignancies with these 7q deletions performed by Michelle and Janet Rowley and their colleagues to find these commonly deleted segments within chromosome band 7q22. And again, while the Human Genome Project was ongoing, we identified and sequenced multiple different candidate genes, and we failed to ever uncover frequent second hit mutations in any gene from this interval or in a second interval that was later defined. And working with Nigel Colleen in the lab, thinking that this might be due to haploids deficiency, Jasmine Wang began using chromosome engineering to generate large synthetic deletions of mouse chromosome 5, 2 intervals defined by Michelle called 5A and 5A3 and 5G2. And we've struggled with this project for most of the last decade. We've made the mice, the alleles work. We've deleted a total of 52 genes, and these mice, frustratingly, still do not develop MDS or AML. And so we were pretty flummoxed and pretty frustrated. Next generation sequencing had come along, and we and others had found that there really isn't a chromosome 7q gene in these patients with monosomy 7 that is affected by second hit mutations. And as often happens in medicine, a group working on an entirely different problem pointed the way to the answer here. And this was a wonderful paper by Nomori et al. That was published in Nature Genetics a few years ago, and doing by a positional cloning and next generation sequencing approach, they identified mutations in a gene called SAMD9 that are germline mutations in a syndrome called Mirage syndrome, and noted in their title that it is associated with the loss of chromosome 7. And the loss of chromosome 7 was occurring in the bone marrows of these patients who were developing myelodysplastic syndrome during the first year or so of life. These patients also had other severe developmental abnormalities, including adrenal gland hypoplasia and severe developmental delay. And SAMD9 and 9L actually turn out to be on chromosome 7, but outside of the commonly deleted regions that we and others had been searching in. However, with all of the next generation sequencing we and others had been doing, we should have found these mutations in the monosomy 7 bone marrow samples we were all trying to study. And so just to summarize then, that 9 and 9L, which is a close homolog, are located at 7q21, proximal to the deleted intervals. These are similar genes. They're upregulated in hematopoietic stem and progenitor cells in response to interferon stimulation and some viral infections. And this shuts down the cell division cycle in these hematopoietic stem and progenitor cells during inflammatory stress and likely protects them from damage. But the stunning observation was that the bone marrow, the monosomy 7 bone marrows of patients with Mirage syndrome actually lost the mutants SAMD9 and 9L allele. So not what we would have expected from the tumor suppressor model. And it turns out that these are actual gain of function mutations. And what happens when there's inflammatory stress in the bone marrow of these patients, it shuts down their hematopoietic compartment. They became severely aplastic, and then a clone emerges that deletes this allele and can then grow out. And so we then went back with hindsight to the data from 25 years before, and this is again this family 3. And when we then looked at the mapping, what we could see now was that in fact these 2 siblings who were affected were inheriting and retaining different parental chromosomes, but they were from the same parent. So it was different parental, in this case, the mother's chromosome 7, but it was the same parent. And of course, the mother didn't matter in terms of passing the disease on. It was the father's chromosome that carried the mutation, and it was the father's allele that was being lost. So it was irrelevant which of the parental chromosome homologs was retained. One other thing we're able to do with those archive samples was to look at secondary cooperating mutations, and they turned out to be quite interesting in that they reflected the same kinds of secondary cooperating mutations that are seen in patients, very elderly patients with MDS who develop monosomy 7, and also in patients who have aplastic anemia and develop monosomy 7. And these include the ETV gene, which is located on chromosome 12p13, and also CETBP1, some RAS pathway genes as well. And this was work that Jasmine Wang in my lab did with Jeff Klo at St. Jude. This is another family, and again, just shows you the heterogeneity. This is a large Mennonite kindred from Canada that we collected over many years. And all of these siblings, so all 8 of them, all developed at one time in their lives. They developed, they developed clinical signs of bone marrow cytopenias. 4 of them actually had monosomy 7. Those are shown in black. They all had the same R1281K mutation, which their mother, who was perfectly healthy, had. And as Ophir told you, when we sequenced these familial bone marrow monosomy 7 patients, we identified a unique group of germline SAMD9 and 9L mutations, as well as the cooperating mutations that drove the progression to MDS and AML. And this is a picture of Jasmine from the UCSF website after this paper came out. So what we know about SAMD9 and 9L is that monosomy 7 clones in children with germline mutations emerge as an adaptive response to assess excessive pathway activation. And this has been termed adaptation by aneuploidy. And it was something that didn't exist in cancer genetics before this particular set of mutations, something we weren't looking for and hadn't thought about, and sort of bad on us for not thinking about this sooner. The hematologic abnormalities resolve in some patients and progress to MDS in others. We know that cooperating somatic mutations overlap with those in other myeloid malignancies with the monosomy 7, and that the parents frequently have normal blood cell counts, and they often have developed somatic mosaicism in their hematopoietic compartment, where they've deleted the mutant SAMD9 or 9L allele and duplicated the wild type allele or the normal allele inherited from the other parent. So they are carriers of this mutation, but in their bone marrow, you sometimes even can't detect it because of these somatic uniparental disomy events. And this is the working model for this. We think that in the presence of inflammatory stress in these patients, it promotes tremendous selective pressure for the emergence of a clone with monosomy 7. And I think 2 things can happen. If there's severe damage to the hematopoietic stem cell compartment as a result of the stress or the nature of the mutation, this monosomy 7 clone is essentially all that the patients have left. And then they acquire cooperating mutations on top of the monosomy 7 clone and progress to MDS and AML. If, on the other hand, they have a reserve of normal hematopoietic stem cells and the inflammatory stress resolves, then these normal stem cells can reassert themselves, and or they can repair themselves through a uniparental disomy event, as we see in the parents, and resume normal hematopoiesis. So this kind of summarizes, this kind of fellowship and beyond, this sojourn around monosomy 7. We were interested initially in the genetics of familial monosomy 7. We began to make and characterize these mouse models of 7q22 deletions in collaboration with Michelle and with Nigel. We also were cloning and interrogating candidate 7q22 tumor suppressors, again in collaboration with Michelle's group. We ultimately identified these germline SAMD9 and 9L mutations as a cause of this familial monosomy 7 syndrome. And now we're kind of closing this loop in a backwards way by going back to our mouse models and breeding them with mice created in Japan that actually lack the mouse homologue, which is SAMD9L. There's no SAMD9 gene in mice, to see if in fact the real cooperativity here is dependent on loss of one allele of SAMD9 or 9L to then favor the outgrowth of these abnormal clones. Now, pretty early on, when we got this disappointing result about this not behaving the way we expected as a tumor suppressor, and given where the technology was in the late 1980s for trying to clone genes, particularly in these very small kindreds, I began to think about maybe I should also think about something different to be studying rather than betting my whole career on the genetics of familial monosomy 7. And something different came along, and I remember this very clearly because it was on St. Patrick's Day in 1987 that a pediatric hematologist in Minneapolis named Jack Priest called me up. And he said, I heard your talk at the Children's Oncology group about monosomy 7. And I have this interesting patient who has monosomy 7, but he also has neurofibromatosis. What do you think about that? And I said, well, I think that's pretty interesting, to have a genetic disease and to have this monosomy 7. And we already kind of knew that monosomy 7 may be sort of a cooperating type event or a haploinsufficient event. And of course, when you have, like me, spent 12 years in Catholic school, including 4 in a Jesuit high school, and somebody named Priest contacts you on St. Patrick's Day, given my Irish heritage, I thought, well, this has got to be some kind of a sign that I should start to study this. And so I did. And this association with JMML and neurofibromatosis had been observed, and I had actually written an editorial about this, so I was aware of it. And JML is an MPN of young children, and these patients have high white blood counts and with dysplasia, so it's an MDS kind of disorder, anemia, low platelet counts, they have splenomegaly, and they have infiltration of other organs. This is one of the few pediatric leukemias that doesn't do very well with chemotherapy and is still only cured with bone marrow transplant, and then only about half the time. There's a peculiar in vitro hypersensitivity to the growth factor, cytokine growth factor GM-CSF in these methylcellulose colony forming assays that I learned with Harold Koenig. And there was anecdotal evidence back in the late 1980s that children with NF1 and with Noonan syndrome were predisposed to JMML. This is what a patient with JMML looks like. They're really sad. This child's belly is massively enlarged because of the infiltration of liver and spleen with these abnormal cells. They often have lung infiltration as well and skin infiltration, and they're just miserable. And it's really tragic how little we have been able to do for them over the years. Now, NF1, as Ophir mentioned, is one of the most common genetic disorders in humans. It's a dominant disease. Affected persons show these pigmented lesions like these cafe au lait nodules, wish nodules in the eye, axillary freckling. And they are predisposed mostly to benign tumors, but sometimes to malignant tumors. The associated cancers are usually derived or occur in cells derived from the embryonic neural crest. And so JMML is an outlier there. And the NF1 gene was cloned in 1991 by Francis Collins and Ray White independently. Ray White, of course, was a former faculty member here at UCSF, and Francis is now the director of the NIH. And when they looked at the sequence of that gene, it was shown to have homology to a group of proteins called GTPase activating proteins, or GAPs. And these proteins were known from studies in yeast to be negative regulators of RAS signaling. And very quickly, Julian Downward, Doug Lowey, and Frank McCormick showed that, in fact, the GTPase activating domain of this NF1 protein, which is called neurofibromin, actually was active against human RAS genes. Now, so this knowledge then sort of catapulted me into a direction that I had never anticipated I would go into, which was learning about this protein called RAS. And so as many of you know, RAS is one of the most commonly mutated proteins in human cancer, and it's a molecular switch that cells use to make decisions all the time. And it's a nodal protein involved in signal transduction that transmits information from outside the cell to the nucleus. And in response to a group of proteins called guanine nucleotide exchange factors, normal RAS proteins dissociate from guanine nucleotide, and because GTP is much more abundant in cells, they assume this active GTP-bound conformation, which stabilizes the tertiary structure of the proteins and allows it to interact productively with downstream effector pathways, such as RAF-MAP kinase and PI3 kinase. Now, once you turn a switch like this on in response to growth factors or other stimulations, you need to figure out how to turn it off. Now, RAS itself is an enzyme. It has an intrinsic GTP hydrolysis rate, but it's not a very good enzyme. And these GTPase activating proteins, or GAPs, bind to RAS and greatly accelerate this off reaction and sort of complete the cycle. And so if you think about this, that the NF1 gene encodes one of these gaps, and you think about this as a dominant familial cancer predisposition syndrome, the next logical question was, is this really functioning as a tumor suppressor? With every cell in the body carrying one mutation, and then tumor cells losing the second mutation by a somatic event to unmask, if you will, this loss of function. And this is an experiment that Daryl Miles, a med student in the lab, did. And we did this work all in collaboration with Frank McCormick, who's a great mentor to me during this time in my career. And we found that NF1 was a tumor suppressor. Here's a family. Mother's affected with NF1. The father is unaffected. And the son has both NF1 and developed JMML. And what we see here, using these microsatellite markers within the NF1 gene developed actually in Ray White's lab, is we can resolve mom's two NF1 alleles, is 403 and 395. Dad is homozygous 399. In the normal tissues of this son shown in lane 3, you see dad's 399, mom's 395, which is the mutant allele. And then in all of the different bone marrow fractions, hematopoietic stem cells, progenitors, what we see is loss of the normal allele inherited from the father. And we see this over and over again in the bone marrows of children with JMML who have NF1. And then collaborating with Gideon Bolag, who, as I told you in the beginning, was a young biochemist and a postdoc in Frank's lab, we sort of connected the dots between the GM-CSF hypersensitivity, which we knew from the work of others, activated RAS, and the downstream effector pathway MAP kinase. We collected samples from patients with JMML that were fresh samples that were flown to us in the cockpit of airplanes from around the country, and labeled them and did biochemistry on them, and showed that in fact, the loss of NF1 in these JMML patient samples increased RAS GTP levels and activated the mitogen activated protein kinase pathway. And then Ruby Collar, when she was in the lab, asked the next logical question, which was, we took all of our JMML samples we had collected, did some of them have RAS mutations? And it turned out that many did, and that the ones with RAS mutations were never from children with NF1 and vice versa, which kind of, again, pointed to the idea that this was the critical lesion in this disease. And so over the years, we began to wonder what other RAS pathway genes are mutated in JMML. And of course, in 2021, this would be a trivial question. One would carry your DNA samples to a core facility, and within a few days, you would have the answer back from a whole genome sequence or a whole exome sequence as to what particular point mutations might be candidates and present in those samples. But this was, again, in the late 1980s and late 1990s, early 2000s. And so what happened? Oh, I'm sorry, I'm going backwards, sorry. And so what did we do? Well, we went back to the clinic and we learned from the patients. So in addition to NF1, Noonan syndrome is a common dominant genetic disorder. It's characterized by dysmorphism and cardiac defects. It was known that these kids with Noonan's will sometimes develop transient myeloproliferation, and that some actually go on and develop JMML. And the break in this story was actually done by a pediatric cardiologist named Bruce Gelb in New York. And Bruce, using standard positional cloning methods, identified mutations in a gene called PTPN11 in about 50% of children with Noonan syndrome. And PTPN11 was known from the work of Ben Neal and others to include a protein tyrosine phosphatase called SHP2. And SHP2 relays signals from activated growth factor receptors to RAS and other effectors. So generally, phosphatases are thought of as negative regulators of signal transduction, but PTPN11 is actually a positive effector downstream of many cytokine growth factors and growth factor receptors. And so when Mignon Lo was in the lab, she pulled out our JMML patient samples and very carefully sequenced them and identified recurring mutations in the NSH2 and CSH2 domains of SHP2, and these domains normally, particularly in the C76K and D50, D59, D51, 61Y amino acids. And you can see that these are key contact points between these inhibitory SH2 domains and the protein tyrosine phosphatase domain of the protein, which is shown over here, kind of the business end and the catalytic domain. And so in the presence of these mutations, the NSH2 domain is pushed away and the phosphatase becomes constitutively active. And then Mignon did these genetic studies, and then Suzanne Schubert, then a grad student in the lab, did a lot of functional and biochemical studies of these proteins as well. So this was good, all was good. We started off studying NF1 and JMML, and I was then again back to the physician scientist dilemma. Were there new lab questions we could address? Should we translate, or should we move into something different? And at this stage in my career, we were pretty interested in this avenue of investigation, and things were going pretty well. And so the question then became, we were going to do more, but the question was really, are we going to now pursue the human samples? And these were very difficult and rare samples to come by, to try to learn from the patients more, or were we going to try to build some model systems where we might be able to attack these questions in a somewhat more controlled environment? And I was fortunate, and then Mignon was really excited about the human genetics and following up on the human aspects of this work, and has done terrific work, including defining her own JMML gene Sybil in her subsequent work. And I became interested in thinking about other models, and this was guided by some conversations with Tyra Jacks. And we decided to try to build some mouse models of JMML. And so collaborating with Tyler and with Luis Parada, who both generously had sent us alleles that they had generated. So Luis had generated a conditional mutant allele of NF1, it's a conditional knockout allele. And Tyler's lab had generated conditional activating oncogenic alleles of the two most common KRAS mutations, first KRAS G12D and then NRAS G12D. This allele, the G12D allele, was generated by Dave Tuveson in Tyler's lab, and this allele, the NRAS G12D, by Kevin Hygas. The Parada and Jacks labs were kind enough to send us these mice before they were actually published, and we mated them with MX1 Cre, which is a conditional Cre that's very active in the hematopoietic compartment. And in every case, we developed, these mice developed a JMML-like myeloproliferative disorder. And interestingly enough, they aren't completely overlapping. The KRAS disease is quite aggressive. The NF1 is intermediate, and the NRAS disease is quite indolent. And the NRAS disease is also more variable. And this is kind of interesting from the point of leukemia biology, since NRAS is by far the most common RAS mutation found in leukemia. And I remember very clearly some experiments that we did, Ben Braun did and Doan Lay did, where we just took the cells from these mice, put them into our standard methylcellulose culture systems that we use to culture JMML cells. And sure enough, they show this same GM-CSF hypersensitivity that the patient cells showed, with the KRAS cells showing greater hypersensitivity and actually forming colonies in the absence of any cytokines, whereas the NF1 cells were simply more sensitive, but did not form spontaneous colonies. Again, this correlated with the more severe disease in the KRAS mice versus the NF1 mice. And these colonies were also giant and monocytic and very abnormal. And I remember seeing these data and thinking to myself, wow, you know, here we can now link a disease, a complex human disease phenotype, to a single gene mutation. We had no idea back at this time what other mutations might be present in JMML, but based on these data, we were pretty sure that it was these RAS pathway mutations that were actually causing a lot of the disease we were seeing in these patients. Now, one of the holy grails for many of us, and I should just call out the remarkable work of Kevin Shannon, targeting KRAS G12C. But RAS is kind of like an addictive drug. Once you get into the RAS sort of cult, you start thinking about why this little 21 kilodalton protein causes so much misery and why we know so much about it and we still can't make drugs to treat these patients. So this is RAS. Here's the gamma phosphate coming into what's called the phosphate binding or P loop of the protein. And here's the arginine finger of GAP coming down to enhance the hydrolysis. And diabolically, the three common mutations that are found in human cancer are at glycines 12 and 13 and glutamine 61. And they disrupt this interaction between between the GAP and the gamma phosphate and greatly impair the off reaction and leave these switch 1 and switch 2 mutations engaged with effectors and activated. And this has been, this used to be called undruggable. We don't think that anymore, but it's one of the most challenging problems in cancer drug discovery. And given that, many companies have thought about, well, let's target downstream some of the kinase effector pathways. And the two most important ones in cancer are the PI3 kinase AKT pathway and the MAP kinase RAF-MEK-ERK pathway. And we've worked extensively with this particular pathway in our myeloid leukemia models and with a drug called PD901, which is an allosteric mechanism. And this is work Ben Braun did with a medical student, Natalia Lubinska, and Tiffany Chang in the lab subsequently did similar work in our NF1 models. And what these data basically showed is that if you look at white blood cell counts in these mice, you can see in the KRAS mutant mice that are not treated, you get these very high white blood cell counts, and they eventually die from this very relentless and progressive myeloproliferative disorder with severe anemia. When you treat them with this MEK inhibitor, their white counts completely normalize. This is not just an anti-proliferative effect because their hemoglobin values also normalize. And this work has now informed a clinical trial that Mignon and Elliot Stieglitz are running nationally of the MEK inhibitor trimetinib in relapsed and refractory JMML patients. And we expect that trial to read out in the next year or two, but it's been enormously rewarding to see work that we started studying human patient samples to try to understand why these children were developing this very aggressive and fatal leukemia, going through the mouse models to model the disease so we would have a way to test drugs, testing drugs in those models, and then informing the work by our translational and clinical colleagues to now bring these therapies back to the patients. Okay, so again, we're all good. We're now sort of in this sort of 2005 era. And again, back to the physician scientist dilemma of what do we do now? So there was the question of a new lab question. And with respect to the models that we had built of JMML, Ben Braun and Ching Li in the lab, wonderful trainees, were launching their own labs. We're very interested in studying the stem cell biology and the myeloproliferation and just the sort of kind of getting to the bottom of how RAS actually functions in hematopoietic stem and progenitor cells. And so that was going to be their lab question. That wasn't going to be my question anymore. And the translation, as I told you, was going to be led by Mignon and by Elliot. So it was my turn to try to figure out something different to study. And again, was fortunate to collaborate with a former trainee in the lab, Christian Kratz and Martin Zenker. And after Christian left the lab, he went back to Germany and was involved. He's a pediatric hematologist now, the professor in chief of the Department of Hematology and Oncology in Hanover. Very excellent clinician and very astute. And Christian was performing JMML testing for Europe for the European Working Group on Myelodysplastic Syndrome and Myeloproliferative Disorders. And a female infant presented in Italy. She looked like this with a high white blood cell count, splenomegaly. She had less than 20% blasts. She had pulmonary stenosis, which is a hallmark feature of Noonan syndrome. And she had these Noonan's syndrome facial features. So you can see the low set and posteriorly rotated ears, which Ophir could easily recommend and recognize and diagnose. We suspected she was suspected of having JMML, but she didn't have a PTPN11 mutation, but had an unusual and never described before KRAS mutation in her blood cells. And then Martin Zenker identified additional patients in Germany, including this young man. And so these mutations were really interesting because the cancer mutations are all kind of clustered around these 3 amino acid substitutions. And these were kind of all over the place in the protein. And we reasoned that they might function differently functionally and biochemically. And so we began to study them. And Suzanne Schubert, who was fresh off of her work with studying SHP2 and now knew all the techniques, did this progenitor colony forming assay. And you can see the wild type cells, fetal liver cells infected with a wild type KRAS have this sort of growth pattern. The T58I mutation from the severely affected patient is here. The V14I from the less severely affected, modestly hypersensitive, but nothing nearly as sensitive as oncogenic KRAS. And so these mutations were hypermorphs compared to the normal allele or wild type protein, but hypomorphs compared to the oncogenic allele, as you might expect since they were tolerated in the germline. And then Gideon Bolag pulled his pipette man out again to do some experiments with us. As I said, Gideon's a brilliant RAS biochemist and had been in retirement as the chief scientific officer at Plexicon, but we made some recombinant proteins and did some experiments together. And this is an assay that Gideon invented, an intrinsic GTPase assay. And you can see the wild type protein hydrolyzes, the G12D protein hydrolyzes GTP very slowly, the wild type much better. These 2 mutant proteins, again, right in the middle. And similarly for the GAP assay that Gideon invented, looking over different GAP doses for an 8-minute time course, the oncogenic G12D is dead. The wild type hydrolyzes very well. And the 2 mutant proteins are sitting somewhere in the middle. And we studied many of these proteins, and they have lots of different sort of interesting biochemical and functional properties. The bottom line being that they're invariably activated compared to wild type, but less activated than mutant. And we've got work ongoing with Gideon and Panyu Chen from my lab, or formerly from my lab, to try to tease out some very interesting potential functional phenotypes of some of these mutations. So this kind of gets me to the sort of end of part 1 of this, or part 2, I guess, just kind of underscoring this remarkable sort of overlap of cancer and development in pediatrics and in JMML and in these developmental disorders of the Noonan's family spectrum, which are now called RASopathy disorders. And we didn't know it at the time, but NF1 is the founding disorder, which includes Noonan's and Costello and CFC syndrome, also Leopard syndrome, and a disease I like to call Lowe syndrome that Mignon identified by studying some JMML patients who actually had germline mutations in Sybil. And we now see that many of these JMML and developmental genes that activate and hyperactivate MAP kinase signaling are the same. And I think that this kind of knowledge, as we begin to transition from treating cancers with these kinds of signal transduction inhibitors, may actually bring us to a day when, believe it or not, we actually may be able to treat human genetic diseases with a pill. We may be able to turn down abnormal MAP kinase signaling in some of these patients with these syndromes and improve their neurocognitive outcomes and some of the other complications they have in the same way that we now think about taking cholesterol reducing agents. Okay, so I'm going to try to run through the last 15 minutes or so quickly for the younger audiences, younger people who are logged in. This picture of David Bowie, who died last year, and he had a song in the 1970s called Changes, and the lyric went changes. And so I was back to where I was. Now I'm in the middle of my career, and I'm doing 3 different things, as many physician scientists do. I'm doing some research, I'm doing some administration, and I'm doing some patient care. The research, I was getting better at this, and I now knew it was time to do something a little different, and I knew what that was. I was beginning to become more like our basic scientist colleagues. I'd locked into the RAS pathway and something I really wanted to keep working on, and had some good ideas, I thought, to continue to work on it. I was involved in administration. Frank had appointed me, given me the opportunity to be the program leader in hematologic malignancies in the Cancer Center, which I enjoyed very much. And as Ophir said, I was vice chair for lab research in the Department of Pediatrics. And I was still very much enjoying taking care of patients. And then Art Weiss and Keith Yamamoto offered me another new, interesting opportunity, and that was to become the director of our spectacular medical scientist training program, or MD-PhD program. And I remember being first deeply honored about this and thinking this would be something really fun, and it turned out to be really fun, just an incredible opportunity to work with all the wonderful young scientists and physicians who come to UCSF and help guide them through their 20s. But when I got home and talked to my wife, Beth, about it, she said, well, what are you going to give up? Because you're already way too busy, and now you're going to take on this big administrative role. And I thought about that a lot. And I think ultimately it came down to a decision between making my lab a lot smaller and essentially focusing more narrowly, or reluctantly giving up patient care. And so it was at that point in my career that I stopped seeing patients. It was about 2007. And, you know, I certainly don't regret that choice. I think it's one that many physician scientists make. And I think at that sort of moment, you sort of switch over to being maybe more of a scientist physician. And I still miss it, but it was probably the right thing for me. The idea that I had was to try to now take these mouse models of RAS-driven JMML and try to progress them to more aggressive cancers with multiple mutations. And the idea we had to do that was to use retroviral immunogenesis, which is probably pretty familiar to some of you, but you inject basically a mouse with a virus that doesn't contain any payload, but a viral LTR that inserts in the genome and can deregulate gene expression by either interfering with it or by activating it. You can then clone those integrations to figure out where they've landed, and can actually use something called Southern blotting, which we've used a fair amount in the old days to try to track these clones. And so again, Jennifer Lockley in the lab, Ching Li and Monique Dale, and Ching Li again, working with the virus Linda Wolf provided us and with the strains provided by Luis and Tyler, did these retroviral forward genetic screens. And again, we kind of learned that NF1 and inactivation and KRAS and NRAS activation really aren't the same thing, but they're similar in many ways. Jennifer mostly got AML out of the NF1 screen. Monique and Ching got TALL with some AMLs out of the KRAS screen. And the NRAS bred beautifully and just gave us M4, M5 AML. And these leukemias are clonal, and they're highly stable when you transplant them into recipient mice. And they all have the same sort of set of retroviral integrations that differ from each other. And then we have activated NRAS or KRAS or inactivated NF1. And so this is kind of the scenario that Jennifer Lockley invented to try to use this as a platform for studying drug response and resistance. I told you we either inactivate NF1 or activate KRAS or NRAS before or after injecting the virus. We generally do this afterwards because that seems to be the most common way that it occurs in human leukemias. We then cryopreserve these human leukemias, inject them into what Jennifer calls a factory mouse, and then generate cohorts of animals that are transplanted with the same leukemia, and then treat them with drugs to see if the drugs actually work. And if they do work, do the mice ultimately relapse and develop resistance? What are the clonal dynamics of that relapse, and what's the underlying mechanism? And as Jen said one day, from the clinical trials point of view, this is like having 10 identical twins with the same leukemia come into your clinic on the same day, where you can do these very rigorous binary comparisons of whether a drug works or not and how the cells might evade it. And I decided, we've done a lot of these screens and published a lot of work about this, and I just decided to talk to you about the very first and the very last papers we published. This is Jim's very initial experiment in the NRAS mutant mice. You see this very aggressive leukemia. These mice die by these leukemias. They die after about 2 weeks. And you can see the dramatic response to an early iteration of the MEK inhibitor called CI-1040. And you can see that these mice have a dramatic response, but the white counts eventually come back. You can see their survival is dramatically extended, but they ultimately relapse. And a key question was, are these relapse leukemias the same or different? And we can do this genetically or functionally, but functionally what Jen was able to show is that now, instead of these leukemias being hypersensitive to the drug in methylcellulose colony forming assays, they now had acquired a phenotypic drug resistance. And so we began to think about, well, why is this? And there were 2 general models, again, back in the late 2007-ish. One was that cancer was a clonal disease. You treat it with a drug, a cell sustained mutations, and that off it went. On the other hand, another idea was that, in fact, cancer was not so clonal. We were just seeing the tip of the iceberg, the main clone. There are lots of other clones, and all one was doing with our drug treatment was selecting for a pre-existing clone that might grow out. And interestingly enough, this mouse model gave us one of the clearest demonstrations of that. So here's CI-1040. Here's the other MEK inhibitor that I've told you about, the later generation, PD-901. These are untreated mice, and you can see 3 bands on the southern blot. This is resistant clones that came up from CI-1040. And you can see this new band is here for CI-1040. And you can see resistant clones from 3 independent mice treated with PD-901. And again, here 3 new bands, this new band. And so what this tells you just immediately is that since these are independent mice treated separately, that these integrations, these clones had to be there initially, because each of these southern blot fragments represents a unique place where the virus has landed. And so this virus would have had to landed in the same place 3 different times in 3 different clones to get this outcome. And of course, that's statistically not happening. These clones were rare clones that we couldn't detect early on that ultimately grew out under the selection of MEK inhibitor treatment. Now, one thing we were a little perplexed about was why they looked different, why CI-1040 and PD-901 selected for different clones. PD-901 is a much better drug than CI-1040. It turned out these were 2 different genes in the same protein family member called RAS-GRIP. And the 2 RAS-GRIPs were overexpressed in their guanine nucleotide exchange factors. And we were able to show with urine roost that they increased the output in the pathway and also conferred resistance. So this was really exciting to us and sort of showed that this model was likely to be operative and was very consistent with emerging data that was coming out from now deep genetic sequencing of human leukemias. And so in the next experiment we decided to do, and the most recent one was Annika Wandler very bravely wanted to know, what about a drug that we actually use to treat patients? And the drug that she selected was a steroid called dexamethasone. And steroids are used to treat every lymphoma and lymphoid leukemia. It turned out, perhaps not surprisingly, to be the best drug we've ever studied in this model system. This is a TALL system. And you can see a dramatic increase in survivorship with just single drug treatment, but that all these leukemias ultimately relapse, exactly what you would expect from human patients treated with single agents. If we then take these cells and transplant them, they're now phenotypically resistant. And what Annika discovered that was interesting were decreased expression of the glucocorticoid receptor in about 40% of the leukemias. And about a quarter of those actually had mutations in the gene encoding the glucocorticoid receptor, which is called NR3C1. And in this particular leukemia, we sort of got what we more or less expected. That is, this L282 frameshift mutation grew out in every single mouse that we transplanted with this leukemia, and that the level of allelic expression correlated with the reduction in glucocorticoid protein expression. What was very surprising to us was this K514 mutation, this Q517 mutation, and this I5113 mutation only occurred in single mice of the 10 we had transplanted and treated. So again, this provided fairly strong evidence that these were somatic mutations. We wanted to know if they were actually causing the resistance. We did some CRISPR studies in cell lines, but I personally like this experiment. This is leukemia 20M. You can see the mutant allele is not present in the parental leukemia at detectable levels on the sequencing trace. You can see at resistance, it's about 50% of the alleles. If we then retransplant this leukemia, treat it with vehicle, it stays about 50%. But if we treat these leukemia again, again, expose it to dexamethasone in vivo, we now see rapid selection for the mutant clone, the clone with the mutation, and almost complete in the retreated leukemias, loss of glucocorticoid receptor protein expression. So again, this surprisingly, and again, 10 years out from the first paper, taught us that maybe this model itself is a little bit oversimplified. And in fact, there are drug tolerant clones that survive initial therapy. And these clones are hanging around while patients are in remission. They're not capable of causing relapse until they acquire a new mutation. And when they acquire that new mutation, it drives relapse. And the relevance of this work to human ALL was recently reinforced by our colleagues at St. Jude, who studied a very large population of relapsed pediatric ALL patients and found exactly the same mechanism, and that NR3C1 was among the most common genes that was somatically mutated during therapy for pediatric ALL. So I am going to stop. I was going to show you one more set of experiments, but I'm going to stop. I'm sure everybody is tired. And I'll just quickly go through this, say that we are also working with Ben Cravat's lab in San Diego on the palmitylation pathway. And it's a very exciting new direction for us. And I think now probably what for me is the most important part of the talk is the acknowledgement section. I've already tried to acknowledge people who've done the work, some of our colleagues and collaborators during the talk. Certainly Michelle, Tyler, and Gideon have played a key role in all of this work. So number one, first and foremost, is my wife, Beth, who's been a wonderful, inspiring, loving partner for 40 odd years. I told you that I did my pediatric residency at UT Southwestern. What I didn't tell you is that UT Southwestern was my third choice. My first choice was the Children's Hospital of Philadelphia. My second choice was Boston Children's Hospital. Neither of those residency programs wanted me. And as a result, I went to Dallas, where I met Beth on a blind date. So that was the best thing that could have possibly happened to me. Where I met George Buchanan, who sort of inspired me as a MD only to think about maybe a research career. And it also placed me in the middle of the country so that the Navy was agnostic about whether I went to the East Coast or the West Coast. And when Beth and I decided to apply to come to the West Coast, we were fortunate to be signed to the Bay Area. So this is a picture of us back soon after we moved to San Francisco. This is a more recent picture of her that I really like because it sort of epitomizes her grace and her style and her beauty. And this is her in the backyard with our dog, Pablo, wearing reindeer ears a couple of years ago. It's a picture of my parents at their 50th wedding anniversary. They're both gone now. My mother was the child of Danish immigrants. My grandfather was a carpenter. She went to Cathedral High School in New York, and in 1943 took a job as a secretary at Columbia University. And there her boss told her that she was too smart to be a secretary and should go to college. And so she eventually did that and eventually became a teacher and then a special education teacher. We only learned in the 1970s when we got a letter from the State Department that she had actually been working on the Manhattan Project at Columbia, and that her boss was one of the physicists who was leading that project. My father grew up in the Bronx. My grandfather was a semi-pro fighter and a fireman in Brooklyn. He kind of bounced around like many kids during the Depression, eventually made it to St. Francis College in Brooklyn. And then the war intervened. He finished up after the war, after serving as an ensign in the US Navy in the North Atlantic, came back, finished his degree at Holy Cross College, went to law school at Columbia. Both of my parents then were first generation college graduates and were intent on all of their children being successful. And so if one thinks about giving the UCSF faculty research lecture as winning at some level a very challenging 100-yard dash, my parents made sure that I started on the 50-yard line. And I'm deeply grateful to them for that and for the love and kindness they showed me through my whole life. This is my extended Shannon family, and you'll want to just point out an interesting kind of experiment in complex genetics. Here I am with Beth. So I'm about 5'11", those of you who know me. It's my brother Glenn, who's 19 months younger than me, with his wife Lori, and Glenn's about 6'2½". It's my brother Brian, who's about 2 years younger than Glenn, with his wife Jackie and a couple of their kids over here. Brian's about almost 6'5". And this is my brother David, who's about 2.5 years younger than Brian. Father of the bride, his wife Martha, his daughter Claire. And David's will only claim to be 6'7", but we all think he's a little taller than that. It's my sister Tara in the background with her partner Hal, and various nieces and nephews arrayed around. My family is incredibly supportive and entertaining, and a lot of really wonderful people to be around. And ditto, I also kind of won the lottery with my in-laws. It's a picture of Beth with her brother Peter and her sister Faith, with our first dog Arlo, beloved border terrier. That's a later picture of her parents, who are also both now gone, Faith and Beth and her brother Peter, in San Francisco for Christmas some years later. And this finally is a picture of Peter, his wife Sarah, my niece and nephew Matthew and Hannah. And we've been seeing them for pizza night, every Sunday night for as long as I can remember, certainly since as long as these guys were born, so 25 years, and they've been part of our COVID pod for all this time. These are people from my lab, and I've been really gratified by how happy and supportive they have been, and I asked them to send me some photographs of themselves. One aspect of my lab has been many of the trainees have also started their families while they've been in the lab, and so some of them have also sent pictures of themselves and also of their kids. This is Lucy Side, who did a lot of the NF1 work, and her son Oliver was born in the lab, and her son Guy actually worked in the lab with us last summer. Brian Weiss runs the NF program at University of Cincinnati Children's Hospital. Tomoko Katsumoto with her worked in the lab as a med student with her husband and daughter, wearing her Fauci We Trust in Science t-shirts. Stoanne Lay, who made the NF1 model, and Kenny Liu worked on the Monosomy 7 project. This guy was born when Doanne was in the lab. And this is Doanne, Ben Braun, and Kenny with Michelle Hermiston from our division at a recent American Society of Hematology meeting. It's Mignon with her family. These guys were both born in the lab when they were in the lab, and Jennifer Lockley with her 3 boys and her husband Brent. And here's a picture of this guy and this guy at about the time their moms were working in the lab. That's Christian Kratz with his wife and baby daughter. Ernesto Diaz Flores with his wife and daughter. And a picture of Suzanne Schubert, who did so much of the biochemistry of the Nunez mutations when she was a grad student in the lab. Qing Li and I, in her hometown of Beijing in Tiananmen Square, Picture that Chin just sent me, a holiday picture of her and her husband, son, and daughter. Annabelle was born about a year after Chin joined the lab. Monique Dale, who did some of the work with TALL in the AML, in the TALL model with her husband and son and daughter. This guy was born when she was in the lab. Tiffany Chang. Ari Firestone. I didn't have a chance to talk about Ari's really wonderful work on a palmitoylation cycle in the lab, but you can see that his son here, Helix, is already getting ready to go to grad school or med school. He's got that half-eaten piece of pizza that he grabbed at the end of the seminar that he's chomping on here. It's Mike Burgess and Jin Su, who met each other as trainees in the lab of my good friend Charles Sawyers, and eventually came together by separate paths to San Francisco. Jin was a grad student, Mike an MSTP at UCLA. And this is Jin's girls and her husband recently, and Mike and his family. Annika Wandler, recent and grads from the lab, Annika Wandler, Panyu Chen, and Noemi Zambetti. Some of the current members of the lab have kind of kept up along the same lines in developing both their careers and their family. This is Ben Huang, young pediatric heme-onc faculty member with his wife and daughters, and Anya Levinson, also a young pediatric heme-onc faculty member with her sons and husband. And finally, a socially distanced 66th birthday party for me for the half of the lab or so that were able to come in that day. I've talked a lot about Jasmine Wong, who's worked so tirelessly in the lab for so many years on so many projects, particularly the monosomy 7 work, and many of the other members of the lab who I'm deeply grateful to for all of their contributions over the years. So thank you all for your attention. And again, I'm deeply honored by the Academic Senate and by my peers to have been invited to give this lecture.
Thank you, Kevin. That was a really amazing talk. For those in the audience who want to ask any questions, the Q&A is open. I think you have to put it straight into the Q&A and not into the chat. Maybe while we're waiting to see if there are any audience questions, I'll just start with something that I've been really interested in as I was looking through your papers and thinking about your work, in this idea that you've studied the RAS pathway and RAS activation in cancer, but then you got into quite early working on these conditions in which the RAS pathway was hyperactivated constitutively in the germline patients, and what that must do to the signaling in the cell. And so how do you think about, first of all, these patients and what's going on in all of their cells that are not turning into cancer, and what's keeping that at bay? And then how are their cancers different, having grown up sort of in this environment than a similar oncogenic mutation in a patient without a germline mutation?
Yeah, no, I think those are really good questions. And I think that I think one of the things we're needing to understand is that understanding cancer in the KRAS in the context of cancer has a lot to teach us perhaps about developmental disorders. And then understanding some of these germline mutations really has a lot to teach us about KRAS biology. You know, the cancer-associated somatic mutations are very activated, and they're not particularly subtle as a group. And Neil Rosen in particular has elucidated, you know, a lot of feedback mechanisms that are deployed to sort of try to restrain the output of this pathway. And I think the reason that that so many of these KRAS mutations are tolerated in the germline is that the cells compensate for them in different tissues in different ways. And I think that that sort of give and take between RAS being activated and how the cell sort of responds and acts to turn RAS off, that the germline mutations are going to be way more informative in terms of some of the more nuanced mechanisms that cells use to control RAS signaling than the oncogenic mutations are. I also think there's a really dramatic sort of difference in thinking about these from a therapeutics point of view. In the cancer side, we're basically trying to target a pathway that cells use all the time to do lots of different normal things that's massively overactive in a subpopulation of cancer cells. And so we're trying to, we know we're imposing collateral damage on the normal cells. We're just trying to limit the collateral damage and kill the bad guys, make them go away, if you will. So really a very dramatic cell fate change from proliferating to dying. On the other side, I think in the developmental disorders, it's a very different hypothesis because we're not really trying to kill any of these cells. We're trying to nudge them into sort of a more normal behavior. And my sense is that as we do that and we do it carefully and kind of understand the biochemistry of these drugs in different tissues, what we'll be able to see is that we may be able to prevent or even reverse some of the kind of the abnormal consequences of these developmental disorders at drug doses of these MEK inhibitors or similar drugs that I think may be very well tolerated. The allosteric MEK inhibitors may not be the ideal drugs for this. There are CRAF kinase inhibitors coming along, and there may be more subtle ways of perturbing the RAS-RAF interaction that would be therapeutically viable particularly for the germline mutations. So I actually think that within a few years, 5 years or so, we'll be doing these kinds of clinical trials, and it'll be challenging from an ethical point of view and from a risk-benefit point of view, and it'll be challenging also, Ophir, for geneticists to start thinking like oncologists and running clinical trials and looking at
good
kinds of outcomes. Yeah, it'll be fun. I think we think about genetics as a gene therapy sort of field, but I think the RASopathies sort of open this new avenue of maybe a biochemical and not necessarily replacement biochemistry, but actually modulating the output of the pathway as a therapy.
Yeah, no, I really I like how you frame the difference between the treatment approaches in cancer versus in the germline mutations, because you're right, they are you're looking at it from a different perspective. Kevin, I hope that I'm not doing anything wrong in terms of questions, that there aren't questions that are sitting there that I can't see, but I don't see any open questions. Here we go. Okay, now they're coming through. Maybe they were stuck somewhere. So Deepak Srivastava is asking, would single cell approaches help determine why selected cells have distinct effects?
I'm not sure I understand the question. I mean, I think we've talked some in the lab about single cell approaches for identifying resistant clones, and the problem there is Deepak, I'm sure, realizes from his experience.
Sorry, and Deepak is now adding consequences of mutations, is what he says. Would that be able to help you determine consequences of mutations?
Yeah, I think that's a really good point, and I think that's right. I think some of what we do in these methylcellulose colony assays is we are analyzing single cells. I think single cell biochemistry would be quite interesting, particularly in something like the hematopoietic compartment, or for Deepak's, perspective in different populations of cardiomyocytes. I mean, we do know that in the Noonan's patients, they frequently develop hypertrophic cardiomyopathy, and we know that tracks with certain mutations, particularly BRAF and RIT1. And so it would be interesting if we had biochemical, rather than just genetic or sequencing, but biochemical ways of identifying different subpopulations of hematopoietic stem progenitors or immature and more mature cardiac cells. I do think single cell approaches could be really valuable there. I do think because of the feedback in the RAS pathway, we do know from even trying to study the oncogene mutations biochemically that they have quite subtle effects biochemically. So that is going to be kind of a lift, I think, but it's obviously something important for the future.
Great. And then there's another question here from an anonymous attendee who says, thank you for a very wonderful talk. A general mentorship question. You've worked with researchers in every stage of training and background. Are there patterns or qualities in the most successful researchers that I can share with a college student or a medical student or a resident or fellow? A perennial question. Is there a way to tell or signs for, am I cut out for life in research and picking a clinical versus academic versus industry career? And this is a question the person wants, particularly for the trainees listening.
Okay, so I'm happy to try to answer that. When people ask me that question, I think the thing that the story that always comes to mind is when I was a third-year medical student. As many people in my generation, we spent every third night in the house on our surgery rotation, and we had the beeper until 5 p.m. And if the beeper went off at 4:55 and a patient was in the emergency department, that was your case. You had to go to the OR with that case. And so 90 or 95% of medical students, including myself, if that beeper went off, we'd be thinking, oh, damn it, I'm going to be here for another 4 or 5 hours. Whereas 5 to 10% of my classmates thought, oh, goody, I get to do another case and go back to the OR. And of course, those were the people in my medical school class and every medical school class who become surgeons. They were just absolutely passionate about what they were doing. And I would say the same thing about trainees. What we do as molecular biologists or biochemists or cell biologists is a sort of variation on the same disease. We're just kind of obsessed with this kind of problem solving and answering the question. And I have learned in my life to not sort of think about research as being one kind of research as being better than another. The yeast cell cycle is just as important as cancer therapeutics. You just need to do good research and good science. And I think the number one priority or number one thing about that is that people be passionate about what they're doing. You should be waking up in the middle of the night or early in the morning thinking about the next experiment or the next clinical trial or the next question that you want to answer. I will say from and I think many of the lab PIs would probably agree, is that one of the things that really marks successful lab bench scientists is that they're just fearless and enthusiastic about doing experiments. It still is a trial and error procedure, and people who really like to put their lab coat on and pull their hair back and put a pair of gloves on and actually do experiments and see something nobody's ever seen before. Those are the people who, in my experience, have been the most successful.
And
I don't know if you want to add to that, or any of the other lab scientists want to add to that.
I think only the two of us can talk. Everybody else is on silent mode. But I have one more question here from Barbara Koenig, which is whether you have any advice about how to approach the ethical dilemmas around treating conditions like the RASopathies.
Yeah, I do. Barbara, it's a really great question. And I think, I'm going to say this as a cancer biologist, and that is, when we talk about a patient that we see in our clinic who has, say, relapsed with JMML after a bone marrow transplant and has a median survival of 3 months. That patient, even if that patient is 6 or 8 months old, we think deserves to be on a clinical trial. On the other hand, when we see a young child, maybe the same age, maybe a little older, comes in with pediatric acute lymphoblastic leukemia, the most common leukemia, that child deserves standard of care because there's a 90% chance we're going to cure that child. And so I think what really has to happen in the RASopathy community is there is to really be a really thoughtful discussion about risk and benefit with the patients, with the patients, with their families, and with bioethicists like Barbara and others, to really think about what is the ethical risk benefit of, say, treating a patient with a MEK inhibitor. So there are 2 very interesting case reports that Bruce Gelb has been involved in of 2 young babies with hypertrophic cardiomyopathy. And they were both on a ventilator they both happened to be in Montreal. And they were treated with a MEK inhibitor. Their hypertrophic cardiomyopathy got better. Their cardiac outputs improved. They got off the ventilator when they went home, and they're now doing pretty well. So that's a pretty extreme example where you can justify the risk and benefit. I think the place you really get into problems or challenges is the neurocognitive questions. Clearly, some of these disorders like CFC and Costello's have very severe neurocognitive outcomes. And if we could define mutations that had those poor outcomes, it would then be the question of, could we justify treating those patients? And then what would the endpoints be to know that we were benefiting those patients and not harming them? And as I say, I think ethicists need to be involved in these conversations. I think geneticists who are used to taking care of these patients chronically. And I think probably some oncologists who understand how to use these drugs. I think that this is an area where, for example, our JMML trial, and I say R as the sort of royal R or we, the JMML trial that Mignon and Elliott are running is going to be extremely helpful to the genetics community, because a lot of these kids are young babies. And if they can be treated with MEK inhibitors for a year and a half or more, and don't have any adverse effects on a therapeutic dose, that tells us that maybe we can nudge the needle toward thinking about treating some of these younger children with retinopathies. But clearly a difficult question, and clearly one that is going to require a lot of different stakeholders, and including the parents and parent groups who bear the burden of having to care for these children.
Great. Well, thank you very much, Kevin, for a really fantastic lecture, and thank you everybody for joining. I just want to say in closing that together with Mignon Lowe, Ben Braun, and Jeff Feynman, we all nominated Kevin together. We really appreciate him very much, and want to salute Kevin's remarkable accomplishments as a pediatric physician scientist and as a mentor and a leader at UCSF. So please join me virtually in congratulating Kevin on his accomplishments. And this recording will be on the Academic Senate website within a couple of days. If anybody that missed it, they can log on and watch that. So thank you everybody for joining.